Every day, we experience the world using our senses. We touch and see and hear and taste and smell, and all that information is sent along nerves to the big lump of gray matter in our heads that we call a brain. But why does any of this matter in the first place? Life as we know it is nothing more than our brain's chemical and electrical interpretation of the signals it receives. Your eyes respond to light, your ears to sound waves, your nose to chemicals in the air, but your brain only responds to a converted electrical signal that it receives from your senses. What we perceive as the world around us is, in fact, only what our brains can decode from our sensory information. Allow us to start with a quick overview. The only way for sensations from the outside world to reach your brain is via cells called neurons. Neurons conduct an electrical signal along a long projection to a junction or synapse with another neuron. The electrical signal is known as an action potential, and the long projection along which the signal travels is known as the axon. Groups of neurons bundle together to form nerves, which allow the information to quickly reach your brain. All information from our senses is accumulated by sensory receptors, which are part of a group of nerves collectively called the peripheral nervous system. The peripheral nervous system includes all the nerves in our faces, our arms, our legs, our core, everywhere in fact, but our brain and spinal cord, which form what's called the central nervous system. It's the peripheral nervous system that connects our central nervous system with the rest of our body and the world around it. Sensory nerves send the information you sensed to the spinal cord and brain, where it is processed. Once the central nervous system has processed the information, a response gets sent back out along motor nerves, another part of the peripheral nervous system. And that brings us to our first sense, touch. There are 4 different sensory neuron endings called receptors involved in touch. Pacinian corpuscles, which respond to pressure and vibrations. Meissner's corpuscles, which are sensitive to light touch. Ruffini's endings, which activate in response to slow vibrations. And Merkel's discs, which give information about pressure and texture. These 4 sensory receptors are part of a group called mechanoreceptors, which are activated when the end of the neuron is deformed, sending an action potential, the electrical signaling mechanism, racing to the brain. Free nerve endings are mechanoreceptors that activate in response to severe pressure, heat, inflammation, or chemical stimuli, resulting in a sensation of pain. All in all, these sensory receptors tell the brain that something's there. So, let's say a bug lands on your arm. How do you know it's there? Even a touch as light as a bug's will activate the mechanoreceptors of our sense of touch. The action potential races along the nerve until it reaches the end. The next neuron is so close, less than a hair's width away across the synapse, the junction I mentioned before. But the action potential can't jump that gap. Neurons are like wires. The action potential could only reach the next neuron if they were touching. So then what? We want the signal to get to our brain so we can respond and flick the bug off before it bites us. Here's the tricky part. When the action potential reaches the end of the nerve, chemicals called neurotransmitters are released across that tiny gap, so that a new action potential is created in the next neuron, and the signal can pass to the spinal cord and be fast-tracked to the brain. Now, the brain can be quite organized. All the touch signals from all over the body are sent to one specific region called the somatosensory cortex. Instead of all the touch signals being jumbled up in the brain, our entire body is mapped out in the somatosensory cortex on what's called a somatotopic map, just like this. Here are our feet and hands and face and even throat. Right, you can sense a bug landing on your arm. But what if 2 land right next to each other?
Hello.
How can we tell there was more than one? These touch receptors can discriminate between 2 points in your arm that are only a few centimeters apart, sometimes even less. Even with these amazing touch receptors, maybe you don't notice the bug, or maybe you choose to ignore it, and then it bites you. Now it's got your attention. The pain you feel is recognized by different receptors than touch, transmitted using different nerves, and sent along a different path to the brain. You ever have that knee-jerk reaction when you stub your toe or step on a tack? You pull your foot back before you even think about it. That signal is completely skipping the brain, taking the express route, called the spinal reflex arc, through the spinal cord, then straight back out to your leg, to get away from the cause of pain quickly. It's only after you jump away that you realize you're hurt. The sensation of pain is detected by pain receptors called nociceptors. There are thermal nociceptors that are activated by harmful hot or cold temperatures, chemical nociceptors that are activated by chemical irritants like capsaicin in spicy food, and mechanical nociceptors that are activated by excessive pressure. Some nociceptors can be activated by all three stimulants, and some aren't activated by any at all. This entire complex structure exists so that a pressure, temperature, or chemical stimulant can trigger an action potential to be sent along nerves to reach the brain, where the information can be processed so that we can feel the world around us. There is still so much unknown, so much to learn about touch and pain, and research continues to explore this area. But for now, it's time to move on to the next sense, sight. Now, as human beings, we depend primarily on our sight. None of our other senses give us quite as much information about the world around us. We can see dangers to avoid, food to eat, prizes to collect, all thanks to these two gooey orbs sitting in our skulls that are wired up to our brains. Suppose you see a big tree in front of you. The sense of sight begins simply with light. Tiny particles of light called photons that bounce off the tree and everything else, and are collected by your eyes through a lens, just like you'd find in a telescope or camera. The lens bends the light so that an upside-down image of the tree illuminates the retina at the back of the eyeball. Photoreceptor cells called cones and rods are activated by different types of light. Cones by more intense red, blue, or green light, and rods by less intense light. Unlike the mechanoreceptors in the sense of touch that respond to pressure, rods and cones use proteins called G-protein-coupled receptors that initiate a signal cascade when they detect the right stimulus outside the cell. In short, a photon of light changes the shape of the receptor protein, which affects how many secondary messengers are present in the cell, which then changes the electrical potential, or voltage, of the cell. After the cell voltage decreases enough, the cell can trigger an action potential in the connecting synapse neuron, which travels along connecting neurons to the occipital lobe at the back of the brain. Sight is organized in a very interesting way. The cones and rods in your eyeballs split into two groups, the left visual field and the right visual field. Each eye has rods and cones in both groups. The visual field is the area you can see without moving your eyes. So the right part of each eyeball can see photons from the left visual field, and the left part of each eyeball can see photons from the right visual field. The left and right visual field neurons from one eye bundle together to form the optic nerves, which meet at the optic chiasm. Here, right visual field neurons from both eyes bundle together to reach the left half, or hemisphere, of the brain, and the left visual field neurons from both eyes bundle together to reach the right hemisphere of the brain. So far, the sense of sight seems to be upside down and crisscrossed, but just wait, it gets sorted out once it reaches the brain. After splitting at the optic chiasm, each group of neurons gets organized based on which eye it is from in the lateral geniculate nucleus, or the LGN. From the LGN, the signals finally arrive at the occipital lobe, specifically what is known as layer V1, the primary visual cortex. Here, the eye-specific signals are further split based on the orientation of light. Is it vertical? Is it horizontal? Something in between, maybe. And then the signals are split again by the direction the light moves. Even another signal distinction exists, this one of signals straight from the LGN, depending on the wavelength, or color, of the initial photon. These color detection cells are called blob neurons. But what is even more fascinating is that the cells form what is called a retinotopic map. Neighboring cells in the retina will send signals to neighboring cells in the primary visual cortex. Everything stays organized, even through all the crisscrosses and divvying up. And that tree in front of you? Everything we see is a result of photons of light triggering action potentials, which travel along the optic nerve to the primary visual cortex, that are then analyzed by thousands upon thousands of neurons. All this, just so you can know that the thing standing before you is indeed a leafy green tree moving in the wind with a brown trunk. But our eyes are not foolproof, and here is the fun known as optical illusions. We don't see exactly what is in front of us. Our brain compares signals, contrasting colors, and relative sizes to give us an idea of how much light is around or how far away an object is. One theory is that we make assumptions based on our previous experience with perspective. These unintentional assumptions can be used to trick our brains. Here are some great examples. Are these lines parallel? In fact, they are. They appear angled because the photoreceptors in our eyes are designed to emphasize changes, such as the edge of a block of color. The gray mortar line appears to be a different shade when compared to the black versus white tiles, which would appear jagged, but we know it should be just one line. so our brain compromises by making the line appear angled. Which line is longer? They are actually the same length. The top line appears longer because we are accustomed to a linear perspective, where an object in the distance is bigger than it appears. Are the circles moving? Akiyoshi Kitaoka created this illusion design, which takes advantage of tiny, fast eye movements and blinking to make the geometric snakes appear to move. Try keeping your eyes completely still. Are the snakes still moving? Now that your eyes are sore, let's give them a rest and let our ears take charge. Next up, hearing. Take a moment and pretend you are standing outside on a beautiful spring day. You feel the breeze on your skin and see the trees and fields of grass. But what about the little birds hidden up the tree behind the leaves? You can't feel them that far away, and you can't see them behind all the branches. How do you know they're there? Well, you hear them, of course. All the twittering and singing from the birds produce sound waves that travel through the air to reach your ears and begin the process we call hearing. Sound waves are simply changes in air pressure that we detect with the auditory system. The ear is a beautifully detailed and delicate combination of mechanical, liquid, and cellular systems. In order to understand how the ear lets you hear, you first need to know what the ear really looks like. We've all seen plenty of ears every day, everywhere. Almost every person you meet has two of them. But what you see is only the outer ear, the sound collector called the pinna, which directs sound waves down the auditory canal towards the middle ear. When the sound waves reach the middle ear, they push on the eardrum, or tympanic membrane. The eardrum is the first part of the middle ear. When the eardrum moves, the three little bones connected to it, the malleus, incus, and stapes, also move. This is the mechanical part of the hearing system. The stapes is a little stirrup-like bone that transmits the vibrations from the eardrum to the cochlea, the pea-sized inner ear structure involved in hearing. The inner ear structure looks a bit like a snail. It consists of a couple tubes looping off the body to form the labyrinth, which is involved in balance, and a spiraled straw, that pea-sized cochlea I just mentioned. The cochlea is divided into three chambers by Reissner's membrane and the basilar membrane. For hearing, the basilar membrane is the most important because it contains hair cells, which convert a wave into an electrical signal that the brain can understand. But hair cells don't respond to changes in air pressure. They respond to movement of their cilia connected to the tectoral membrane. When the stapes vibrates on the opening of the cochlea, it sends waves traveling along the length of the fluid-filled spiraled cochlea, moving the basilar membrane, and with it, the hair cells and connected cilia. Just like when you make waves in a pool with your hands, and the kickboard floating on top bobs up and down. Different sounds make different waves, which makes different sections of the basilar membrane vibrate more than others, triggering only certain hair cells. This way, you can tell the difference between a screech and a growl. Remember how the sense of sight was kept very organized all the way through to the brain? Well, the sense of hearing is similar. It's called a tonotopic map, and it begins at the basilar membrane. Different sections of the basilar membrane vibrate more in response to specific sound frequencies because of the shape and stiffness of the membrane. Here, near the opening base of the cochlea, the basilar membrane is narrow and stiff, and vibrates in response to about 16,000 Hz, a very high-pitched sound. Here, at the end apex of the cochlea, the basilar membrane is about 5 times as wide, about 1/100th as stiff as at the base, and vibrates at about 500 Hz, a very low-pitched sound. The hair cells that convert the mechanical vibration of the basilar membrane into an electrical signal are mechanoreceptors, the same group of sensory neurons as the touch receptors. When deformed, the hair cells create an action potential which travels along the auditory nerve to the ventral cochlear nucleus, then to the inferior colliculus, to the medial geniculate nucleus, and finally to the auditory cortex. The auditory cortex is organized according to frequency, just like the basilar membrane. Essentially, hearing is just a specialized form of sensing pressures. Sound waves travel through the auditory canal and push on the bones of the middle ear, which then create waves in the fluid of the inner ear. These waves vibrate part of the basilar membrane, which changes the shape of the hair cells to create an action potential that finally gets sent to the auditory cortex in the brain. So this is great. You can hear the birds in the tree, but how do you know which tree it is? Are they in front of you or off to the side? The brain has devised a way to localize where a sound comes from. Sound waves can only move through the air so fast, at the speed of sound in fact, 340 meters per second. As such, when a sound originates from somewhere off to your side, it takes longer to reach one ear than it does to reach the other ear. The action potentials from each ear are thus generated at different times, and the signals can reach your brain at different times. The brain can process this difference to see which ear heard first, and so tell you where the sound is. Fantastic! We can feel the breeze, see the trees, hear the birds, but what if we want to smell that wonderful springtime air as well? How do we do that? Well, the first step is breathing. Breathing is always good. And now, last but definitely not least, our final two senses of this investigation, taste and smell. These two senses are similar and very connected. Have you noticed that when you're sick and your nose is all clogged up, nothing tastes right? Or when you breathe in a wonderful smell through your nose and mouth, it seems so much richer. That's because both taste and smell depend on the same thing, chemicals, and without both senses, your brain can't fully register flavors. But that is where the similarities end. Taste and smell use different structures, follow different paths to the brain, and in general have different organization. Taste, as we all know, begins at the mouth. You put something on your tongue and can tell what it tastes like. But more than just the tongue informs your brain of different chemical tastes. The palate, pharynx, and epiglottis are also involved, though the tongue is the primary taste organ. It is covered in hundreds of little bumps called papillae, shaped like ridges, pimples, and mushrooms. Each little bump can have up to several hundred taste buds, with each taste bud containing about 100 taste receptor cells. That's upwards of 500,000 taste receptor cells. Just imagine all those different flavors. Unlike the classic picture where different areas of the tongue detect separate tastes, in reality, a single taste bud can have receptors sensitive to all the tastes. The 5 primary tastes are sweet, bitter, salt, sour, and umami, which is the delicious taste from glutamic acid salts like MSG, which is added to meat or Chinese food. Each taste receptor cell is activated by one chemical, but not all are activated by the same mechanism. Salty and sour taste receptor cells are activated when ions pass through channels in the receptor membrane, which releases neurotransmitters to generate an action potential in the synapse neuron. Bitter, sweet, and umami taste receptor cells use G-protein coupled receptors, just like sight, where the chemical binds to the receptor, causing a chain of events within the receptor cell to release neurotransmitters and generate an action potential in the synapse neuron. Nerves from the taste receptor cells, called gustatory axons, all bundle together in the brainstem to synapse within the gustatory nucleus of the medulla, continue along to the ventral posterior medial nucleus of the thalamus, and finally to the primary gustatory cortex. Besides these general ideas, not much is known about the sense of taste. But what is taste without smell? Bland and unappetizing as anyone who has been sick can tell you. The mouth and nasal cavity are connected by the pharynx, which allows chemicals to move to the nasal cavity and be detected by smell receptors called olfactory receptor cells, enhancing the flavor of food. Smell is also one of the most potent senses connected to emotion, memory, and motivation. The olfactory receptor cells are also in use every time you breathe. The air enters our nasal cavity, which passes the chemicals in the air over the cilia, or tendrily-looking projections of the olfactory receptor cells within the olfactory epithelium. Each olfactory receptor cell expresses only one type of receptor protein. The odorant receptor proteins on the olfactory receptor cells are the all-too-familiar G-protein coupled receptors that we discovered in the sense of sight, causing a cascade of events when an odorant comes into contact with a proper olfactory receptor cell and its associated odorant receptor protein. The secondary messengers from the activated G-protein coupled receptor causes a depolarization of the cell membrane, initializing an action potential in the cell. The olfactory receptor cells expressing the same receptor proteins all group together into globs called glomeruli within the olfactory bulb just below the brain. From here, a secondary olfactory nerve travels from the glomerulus and, unlike the other senses, bypasses the thalamus to connect directly with the olfactory cortex in the brain. In summary, taste begins in the mouth and throat, where chemical stimulants at the taste buds trigger an action potential in the gustatory axons, which travels to the gustatory nucleus, then to the ventral posterior medial nucleus of the thalamus, and finally to the gustatory cortex in the brain. Smell, on the other hand, begins with chemical stimulants at the olfactory receptor cells in the olfactory epithelium, which trigger an action potential in the cell that then travels along a secondary olfactory nerve directly to the olfactory cortex. So, congratulations! You can now understand how you experience the world around you through your senses. That covers it, right? Nope, not quite. There are more than just 5 senses. Touch, sight, hearing, taste, and smell are just the beginning. We have already seen a few other senses secreted within these videos. Pain and temperature are each their own sense. Within the inner ear, the labyrinth is the main organ of the vestibular system, which gives us balance and tells us how we move through space. We can sense where our own body is, like when you touch your nose with your eyes closed, which is known as proprioception. And as you are all probably aware by now, we can sense the passage of time. How many more senses can you think of?